Adrian's flying hamster research is going really well. I mean, he's figured out that coat color is determined by a single gene, and the brown phenotype is dominant over the white coat phenotype. Now he's going to turn his attention to ear size, because he's noticed that some of the hamsters from the wild have small ears, and some have large, like Dumbo ears. So based on his previous results, Adrian hypothesizes that a single gene is probably also determining this phenotype, and that he can verify this hypothesis if he can find a 3-to-1 ratio between the two phenotypes. Now, recall that he used big B and little b for coat color, so he decides that he's going to use big E and little e for ear size. So he picks big E to represent small ears and little e to represent large ears. He decides to perform genetic crosses again to confirm his hypothesis. First, he has to establish true breeding strains. So he takes brown hamsters with small ears and white hamsters with large ears and establishes true breeding strains of each. So Adrian takes his brown hamsters with small ears and mates them with his white hamsters with large ears. And that's going to produce double heterozygotes at the F1 generation. So he's going to have big B, big E, little e, for every single progeny in this F1 generation. Since we're talking about a cross with double heterozygotes, what we're monitoring here in the F1 generation is what's called a dihybrid cross. That is, it's a cross between individuals that are heterozygous at two different loci. Adrian expects that his F1 cross will produce a 3-to-1 ratio between the dominant and recessive traits. However, he's perplexed to observe that instead he sees a 9-to-3 to 3-to-1 ratio among 4 different phenotypes. What's going on here? Were the conclusions to his first experiment just flawed? Or is there some other explanation that could possibly reconcile both sets of data? Well, let's see if we can help Adrian figure out what's going on. Adrian recalls that genes are located on specific chromosomes. He also remembers that homologous chromosomes segregate into separate gametes during meiosis. However, there's really no rhyme or reason to predict which homolog a gamete receives. Now, what does this mean for genes that are located on separate chromosomes? Well, since they are located on separate chromosomes, it means that the ear size gene is also segregating independently. Adrian's experiment has demonstrated Mendel's second law. Mendel's second law is also known as the Law of Independent Assortment. It states that the alleles of one gene sort into the gametes independently of the alleles of another. To better understand the laws of independent assortment, let's consider some coin tossing. Okay, do me a favor and pause the video while you obtain a coin. Don't worry, I'll wait for you. Okay, let's consider the example of coat color just alone for a minute. So we know that there are 2 alleles for coat color. We have big B and we have little b. And this is just like our coin, where we have heads or tails. Since there are only 2 possibilities, there's a 1 in 2 chance of getting one of the alleles, say big B, just like there's a 1 in 2 chance of flipping the coin and getting, say, heads. Okay, so if we arbitrarily associate heads with brown and tails with white, we can simulate the random nature of chromosome segregation into gametes. Okay, so let's do this together. Flip your coin and see what allele the first gamete receives. All right, since the law of segregation tells us that the homologs must segregate, if the first gamete gets allele 1, the second gamete has to get allele 2. So in my case, I got big B, so that's going to go into my first gamete, and that means little b is going to go into my second gamete. So now that we've applied the law of segregation to our first gene, let's apply the law of independent assortment to our second gene. Alright, so you may have to pause the video again, but we need to obtain another coin. So for instance, if you used a penny the first time, get a dime, a nickel, or a quarter this time. Okay, so flip the two coins several times and monitor your results. Does the outcome of the first coin influence the outcome of the second? No, right? If I flip my coin and I get tails with my first coin, it doesn't make it any more likely for me to get tails with my second coin. This is the idea of the law of independent assortment. Just like the coins, if two genes are located on different chromosomes, the segregation of one gene will not affect the segregation of the second. That means if we are studying two genes that sort independently, there are 4 equally likely genotypic possibilities for the gametes. Well, tune into our next lesson to see if our new understanding of the law of independent assortment can help us explain all these puzzling flying hamster results that Adrian noticed in this lesson. In summary, a dihybrid cross is a cross between individuals heterozygous at 2 loci. Mendel's second law is also known as the law of independent assortment. The law of independent assortment states that the alleles of one gene sort into the gametes independently of the alleles of another gene.